Hello, everyone. Welcome back to ENTX202. Today, we're going to cover the topic on modifying factors on the metabolism and toxicity. So we have been talking about different metabolic factors, metabolic biotransformations. Now, we're going to see what factors can play a role in affecting the outcome of metabolism and toxicity. So this lecture, we're going to talk about different factors, including chemical factors, genetic factors, in particular polymorphisms of different metabolic enzymes, and we're going to talk about enzyme inhibition and enzyme induction, in particular with respect to P450 enzymes. So start with the first factor that are influencing the metabolism and toxicity. As we have discussed, the lipophilicity is an important factor that affects all aspects of absorption, distribution, metabolism, and excretion, every aspect of the ADME. And lipophilicity as a major factor affecting the cellular uptake as well as the distribution play a fairly important role in affecting the outcome of toxicity. What is shown here using a well-known herbicide in the United States, perhaps the most widely used herbicide, paraquat, in the United States. Paraquat is an important tool for control the weeds in many agriculture and non-agriculture settings. However, there were no homeowner use and no product registered for application in residential areas. This is a pretty prevalent chemical, and there were well-documented studies regarding the toxicity of paraquat. The main target organ for paraquat exposure is that it's usually involved in the lethality of the lung damage. So the damage is observed in the destruction of the type 1 to type 2 alveolar epithelial cells. And this is due in part to the selective uptake of paraquat. This is because in those cells, there's selective transporters that's normally taking up polyamines such as putrescine and spermine. These are biological amines. As you can see here, we're showing a structure of putrescine with two terminal amino group with 4 carbons in between. And this has a molecular length about 6.6 angstroms in the whole length of the molecule. Compared to the native substrate that's taken up by these transporters, you can see paraquat has approximately similar length compared to the native substrate. And that is believed to be responsible for the selective uptake of paraquat in those epithelial cells in lungs. So this is a good example of the structural similarity of the two compounds leading to selective absorption or uptake of the particular molecule. Mechanistically, once the paraquat is uptaken and accumulated in epithelial cells, there are two general pathways over years of study people have demonstrated that related to the toxicity of paraquat. Paraquat can, as the structure shown here, can be reduced by NADPH. This is a well-known cofactor mediated by P450 reductase to undergo this one electron reduction process. Of course, the reaction reduction, the redox reaction, reduction oxidation reaction is always reversible under the right conditions. So the reverse oxidation process can actually induce the formation of superoxide anions in the presence of oxygen. This will lead to the production of these undesired reactive oxygen species in the form of radical. And in turn, this can potentially damage the lipids. And with respect to lipid oxidation and peroxidation, this can lead to serious cascaded reactions to generate additional lipid radicals and subsequent tissue damage. So one of the pathway is, of course, generating reactive oxygen species. At the same time, the second mechanism is believed to be the depletion of NADPH. And this is because the NADPH is involved in many additional cellular metabolism, and the depletion of NADPH may compromise the alveolar cells and reduce the ability to carry out essential biochemical and physiological functions. We'll have a separate lecture designated to reactive oxygen species, and we'll talk more in detail about some of the actions mediated by the reactive oxygen species. But this is a short overview of the paraquat toxicity. And again, going back to chemical factors, this is mainly led by the structural similarity of paraquat to the native substrate of the ion transporter that result in the selective uptake of paraquat in the lung cells. In addition, other chemical factors such as the molecular shape, this in a way affect many of the chemical as well as the uptake properties. For example, the well-known aryl hydrocarbon receptor. This is a molecule that recognizes a series of polycyclic aromatic hydrocarbons as well as the polychlorinated hydrocarbons, and in particular dioxin. So these receptors actually selectively recognize the planar molecule, but not those non-planar molecules. In this case, we're showing a molecule with two rings with 6-chlorine. This is called hexachlorophenol. In this case, the hydrocarbon receptor will actually bind to the planar form of this molecule, but does not bind to the form that I'm circling here, that with the angle. So this is the planar molecule bind to HR, and this is the molecule with a specific stereo rearrangement that doesn't fit the aryl hydrocarbon receptor binding. So this is how stereochemistry affects the binding of the target molecules. Another example is the selectively, again, the stereochemistry that result in the selective absorption of the molecules such as L-DOPA to specific from the GI tract, and the L form of the L-DOPA is more prevalent than its D counterpart. The composition and the stereochemistry of the chemicals also play a very important role in bioactivation and subsequent toxicity. The picture showing you a series of compounds here. These are called polycyclic aromatic hydrocarbon, PAH for short. These are a class of chemical that occur naturally in coal, crude oil, and gasoline. They also can result from high temperature combustion of the coal, oil, and gas, and wood, and garbage, tobacco. So many of these PAH are well-known human carcinogens, and we're showing you a few examples of these carcinogenic PAH. And the bottom one is the well-known benzoyl A pyrene molecule with 5 ring here, and the one above it has 4-ring with specific stereochemistry arrangement. In general, we can classify these molecules, pH molecule, into 2 general categories based on the stereo arrangement. One of them we call the Bay region compounds with larger opening in here, as you can see illustrated using the arrow here. So these are called Bay region compound. On the other hand, the top 3 compounds with a smaller opening are known as what we call the Floyd region compound. It turns out the bioactivation of these compounds, based on their stereo arrangement, occur primarily at these labeled carbons. You can see for benzoylpyrene, this is 7, 8, 9, 10. And for BCPH, this is 4, 1, 2, 3, 4 illustrated here. And the top one, 11 to 13, 14 positions. And the bioactivation of this follow the general pattern that we saw before, forming epoxide. And you can see here the general structure, showing this as a structural class. The bioactivation of this compound mainly goes through the epoxide formation with different stereochemistry occurring at different sites. And subsequently, once the epoxide form, we know this can be detoxified by GSH for these epoxide, or followed by spontaneous hydrolysis of those epoxide to form diols, or mediated by epoxide hydrolase. The stereochemistry of many of these products are actually dependent, differ based on different compounds, and differ based on the different stereochemistry of the compounds. The relative distribution of those diol product, as well as the diol epoxide product, are also different under different contexts. As a result, there are different DNA adduct formation or different stereochemistry as well on the DNA conjugate level. So once the DNA adduct is formed, you can see here, this column illustrate the various kinds of DNA adduct in terms of their stereochemistry, in their accommodation and their conformation of these DNA stereochemistry of these DNA adducts, as well as their conformation within the double-stranded DNA helix after adduction. This can vary dramatically based on the stereochemistry of this compound as well. And henceforth, the outcome of repair and the carcinogenicity are quite different based on different compounds. And just to give you a flavor of the varying degree of carcinogenicity, the compound showing on top here, DBALP, this compound is recognized as the most potent tumorigen among all the carcinogenic PAH tested to date. In terms of the carcinogenic potency, this is about 100 times greater than that of benzoylpyrene, one on the bottom here. So that really speaks of the chemical difference as well as stereochemistry in terms of how they are impacting the degree of metabolic activation as well as the toxicity. Moving on to genetic factors. The inherited difference in metabolism distribution of foreign compounds has been well recognized for many years. And this is believed to be an important factor in individual variation in toxicity response. Now, in terms of genetic factor, we know individuals are different on the genetic level, and it's not too difficult to imagine in terms of metabolic activation and toxic response. Many can vary based on the different individual genetic factors. One important term in this case we want to talk about is called polymorphism. This is referred to the occurrence of genetic variants in a population, whereby the occurrence is more frequent than should accounted by new mutation, the spontaneous mutation. Usually a good rule of thumb or a guide in terms of defining genetic population or genetic polymorphism is that the particular variant has to exist in at least 1% of that population of study. So in other words, if you're talking about a single nucleotide mutation of some gene, let's say P450 gene, in order for that mutation to be considered as polymorphism in human population, that the occurrence frequency of that particular variant has to be at least 1% of the studied population. And it has been estimated that about 10% of any given person's enzyme show a significant degree of heterogeneity. And this speaks of the ubiquitous nature of the genetic factors affecting the human metabolism and toxicity. The phenotypic impact of polymorphism vary according to their gene and position of the variant residue. As you can imagine, if it occurs at critical regions of a particular protein or enzyme, it could impact the function, whereas if it occurs at non-essential regions, it could lead to no impact, etc. So we'll see some specific examples. I kind of alluded to this term earlier. So one of the important terms in here is called single nucleotide polymorphism or single nucleotide substitution. So this is most common form of polymorphism. So just a varying by one nucleotide. And this could lead to change of the amino acid of the encoded protein. There are other type of sequence variations besides the single nucleotide variation. There can be insertion or deletion of nucleotide sequences. There could be gene copy variations, for example, duplications. There could also be gene conversions, which are all much more severe and much more dramatic compared to single nucleotide polymorphisms. But SNPs are considered most commonly occurring genetic factors in terms of affecting the metabolic profile and toxicity profile. Genetic factors can affect the toxicity of a foreign compound in two general ways. One is to influence the response of the compound, and the other is to affect disposition of the compound. Genetic factor also believed to be an important contributor of what we call the idiosyncratic drug reactions. These are basically reactions that undesired side reactions that occur in a small population, and there are certain drugs fail due to the idiosyncratic drug reactions, and they do not necessarily follow a parent dose response relationship. So that's why it's oftentimes it's hard to predict in the drug development and testing stage. Historically, one of the most well-studied and most characterized example of pharmacogenetic variation in terms of polymorphism is the N-acetyltransferase gene, NAT gene. And this is first recognized with the use of isoniazid, we talked about this before, as a tuberculosis drug back in the 1950s. Back then, isoniazid was discovered as a highly effective treatment for tuberculosis. Soon after the introduction of this compound, clinicians noticed that some patients receiving the drug developed peripheral neuropathy. This is basically a toxic response characterized by tingling and numbing of the fingers and toes. Hepatotoxicity is another serious side effect from this tuberculosis drug. And typically, when we talk about distribution, if you look at this graph, this is describing the plasma concentration of this isoniazid drug versus the number of individuals. In terms of distribution, normally we will see a normal or log normal distribution. But in this case, isoniazid historically has shown a bimodal, what we call a bimodal distribution, basically two peaks, right? So there's a group of people showing rapid, showing as a rapid metabolism of isoniazid, which result in a lower concentration of isoniazid in the plasma. On the other hand, there are populations that show a relatively normal distribution. So you will see two peaks in this distribution diagram. Subsequent studies over the years have identified the family degree that this phenotype was an inherited Mendelian trait determined by a single autosomal gene, and later identified as an N-acetyltransferase. And this is one of the well-studied and first well-studied and well-recognized example of the genetic factor affecting the drug metabolism. Mechanistically, you can see NAT is involved in the metabolism of this isoniazid drug, showing using INH here. There are several pathways that's considered as a detoxification pathway, and there are pathways such as the formation of hydrazine. This is considered and subsequent formation of the acetyl as a bioactivation pathway. As you can see, the isoniazid, this is what we call a hydrazide group, and this is a hydrazine functionality. This is a classic substrate of N-acetyltransferase enzyme. Metabolically, N-acetyltransferase, NAT2 in this case, can conjugate an acetyl group to the compound. Now, this is considered as a deactivation pathway or detoxification pathway, reducing the concentration of the parent compound. Alternatively, if the compound undergo reactions by amidase, the bond here will break, and the reaction will lead to the release of hydrazine, basically NH2, NH2. And this compound can be further metabolized. This is also a typical substrate of NAT2, can be acetylated here to convert it to acetyl-hydrazine. And this is considered as a bioactivation pathway because this can further undergo the oxidation reaction by P452E1 to form hydroxyamines. And this is illustrated by reactive metabolite here and reactive metabolite here as well. The formation of acetylhydrazine can be detoxified by NAT as well, showing in this pathway. This is considered a detoxifying pathway by forming this diacetylhydrazine. So you can see the NAT enzyme, because its involvement in both the bioactivation and detoxification, and the readily metabolic reaction occur mediated by this enzyme. The NAT enzyme will have a direct impact in terms of the remaining level of isoniazid in the blood. And henceforth, this enzyme, whether individual variation of this enzyme in terms of their the rate of metabolism can directly impact the remaining or plasma concentration of isoniazid that the family in individuals. Over the years, there has been international studying group working on the polymorphism of various enzymes, and NAT definitely play a very important role historically, and there's well-documented individual polymorphisms on various forms of NAT enzymes that exist in the population. Of course, if we call it, remember that threshold, in order to be called polymorphisms, we have to have more than 1% of that occurrence, frequency of occurrence of a particular variant in the population. So if we studied those single nucleotide polymorphisms and then look at it in the context of the 3D protein structure of NAT, as well as the primary sequence, amino acid sequence of that particular protein enzyme, you can see this top panel basically mapped all those genetic variations of the particular amino acid residue on the 3D protein structure for NAT1 as well as the NAT2. The bottom panel, on the other hand, shows you the particular amino acid changes with respect to individual variation. You can see sometimes you will have this variation forming a stop codon instead of arginine in that position, which in turn will lead to a dysfunctional protein because essentially you are chopped the protein into a non-functional fragments. Other times, you will also see variations in terms of single amino acid mutation converting arginine to tryptophan to other residue in other cases as well. And there are many functional studies I follow up on these single nucleotide polymorphisms, isoform of the enzyme, and there are detailed biochemical studies showing the impact of those amino acid change with respect to each SNPs. Just to give you a flavor of that, you can see here a substitution of arginine 64, which is here. It's showing the primary structure, arginine 64, with tryptophan or glutamine will destroy these interactions, leading to poor solubility of the protein as well as protein aggregation. Essentially, you will reduce the availability of the functional enzymes. Polymorphism is also playing a fairly important role in carcinogen bioactivation. And again, here we're showing you some of the well-documented carcinogen known as heterocyclic amines, IQ or PHIP. And these are well-documented carcinogen present in overcooked meat, tobacco smoke, and certain air pollutants. These serine compounds can undergo cytochrome P450 activation, again, because of the formation of hydroxyamine here. And that hydroxyamine can undergo metabolic reactions by sulfur transferase or an acetyltransferase to form sulfonate conjugates as well as acetyl conjugates. As we discussed last lecture, because of the unstable nature of some of these bioconjugates, sometimes they will be hydrolyzed further and then reverted back to the non-conjugated version. In this case, the outcome of that will actually lead to the reaction, the formation of so-called neutrinium reactive intermediate that subsequently cause the biomolecule binding, such as DNA adduct. It turns out that the bioactivation of these compounds primarily rely on P450-1A2. And because the individual difference of cytochrome P450 enzymes, there's also well-documented difference in terms of 1A2 metabolism of these carcinogens. You can see here the top graph illustrated the 1A2 expression of different human individual in the liver. And we certainly have populations where individuals have relatively low expression around 50, a larger population accumulating in this region, right? And there are also individuals that vary quite dramatically that express a high, very high level of P450s up to, this is the number of individuals, and up to 100, 150, all the way to 250, right? So the x-axis is actually the picomole per milligram of protein, and the y-axis is the number of individuals. So you can see distribution-wise, we have a relatively wide distribution, and the individual variability can dramatically influence the 1A2 enzyme. And on the bottom graph here, you can see in terms of the level of, this is a correlation between the level of 1A2 enzyme with the corresponding MEQI N-oxidation product, as well as PHIP N-oxidation product, basically the hydroxyamine metabolite by 1A2 specifically. And we can see, based on the individual variation of P450-1A2 protein level, we can see a generally good correlation between the available enzyme and the outcome, the amount of reactive metabolite in the form of an hydroxylation reaction. In addition to carcinogen bioactivation, we can also see that P450-2D6, this is historically been recognized due to its importance of its role in drug metabolism. And historically, 2D6 has been well studied in terms of the polymorphism because it's recognized the role in impacting the cardiovascular drugs such as debrasquine or spartin, those cardiovascular drugs. In this case, historically, they're similar to the case of NAT enzyme. There were studies classifying different cytochrome P450-2D6 alleles in the population as poor metabolizers, intermediate metabolizers, normal metabolizers, as neutral metabolizers, based on their rate of metabolizing these cardiovascular drugs. Individual with poor metabolizers, individual classified into the poor metabolizer category, they can have phenotypes that possess G to A transition mutation at an intron to exon boundary that describe the splicing of mRNA transcripts. this actually impair the production of functional 2D6 at the MR level. In turn, these individuals have been shown to have a sufficient level of 2D6 protein for metabolic reactions. And that's why they are considered poor metabolizers. On the other hand, individuals within the intermediate metabolizer group, they sometimes involve mutations within the protein coding sequence, leading to enzymes with altered enzymatic activity towards some of the studied drugs. On the other hand, for fast ultra metabolizers, they often found to be having what is called a gene duplication, with 13 copies of P452D6 gene observed in some member of the Swedish family historically. And a recent large study in the US also identified the ultra metabolizer genotype around 1%, 1.5% of the study population. The metabolic difference also manifested in different ethnic groups. For example, 2D6 polymorphism was found to vary its prevalence in different ethnic groups. It's perhaps more frequent in Caucasian population, while the incidence is relatively low in Asian population, as shown in the second point here. And this is very much related to the development of individual personalized medicine. Although the promise of personalized medicine was very high initially, nowadays the clinical opinion seems to be divided concerning the actual benefits, such as diagnostic approaches bring it to the patient care. And these individual differences in terms of polymorphisms highlights the challenge of metabolic studying or understanding completely the metabolic difference between individuals in the population, and also highlight some of the important challenge in bringing the personal medicine into patient care. Next, we'll move on to a different factor, talking about enzyme induction. The induction of enzyme, this is describing the effect of some metabolic enzyme in terms of activity can be elevated. The amount of enzymes increased, but the inducer may cause other changes, such as increase in the bile and blood flow in the smooth endoplasmic reticulum. A signature characteristic of enzyme induction is that induction often requires repeated exposure. In other words, it's not single dose that causes enzyme induction. Oftentimes, it's chronic exposure that lead to enzyme induction. If you look at the table here, we have several good examples of enzyme that can be induced by different chemicals. For example, the well-known inducer of cytochrome P451A2, 1A1, this is dioxin. We also know dioxin is a pretty prevalent toxin. And phenobarbital, this is a drug that used to treat or prevent seizures, and also used as a short-term sedative to help patients relax. This is a well-known inducer of cytochrome P2B family enzymes. And isoniazid, the tuberculosis drug we talked about before, and as well as ethanol. Those are well-known inducers of cytochrome 2E, 2E1 enzyme. The induction of P450 has been demonstrated in many different species, including humans, and in various different tissues, including liver. As I said, the induction usually results from repeated or chronic exposure, although the exposure may vary based on the individual cases. The effects of enzyme induction on the metabolism, this can have various effects based on the extent of a particular enzyme involvement in the metabolism. If the induced enzymes is involved in the metabolism of one foreign compound, this can lead to the increase of rate of the metabolism in that particular pathway. If the induced enzyme is involved in a series of pathways, this can also easily alter the metabolic profile of a particular compound. Of course, if the enzyme is not involved, there can be no effect for that particular metabolic pathway. The table shown below shows some good examples of the change of different metabolic profile under the induction. You can see here the first row here showing uninduced reaction, uninduced meaning that without inducer. And this is showing using an example of warfarin. This is a blood thinner. Warfarin's metabolism and the relative proportion of these R and S isomer formation. Without induction, you can see this is 0.24, 0.22, 0.04, and around a 4-to-1 ratio in both 5-to-1, 4-to-1 ratio in both cases for each isomer. Let's look at under induction, right? So there are two chemicals that have been studied to induce the P450 enzyme. And in the first case, phenobarbital, you can see the induction led to the general increase of overall metabolic formation. And that is consistent with the effect of enzyme induction, right? So we generally see an increase of metabolic rate. And you can see here, the number are all greater than the case we see in the uninduced reaction. In addition, we will see a slight change in the profile. You can see this is perhaps slightly elevated in terms of 7-hydroxy compared to 8-hydroxy. The relative proportion is also slightly increased, although it's not dramatic. On the other hand, you can see the third line here. This is with another inducer. The relative profile of 7-hydroxy and 8-hydroxy products actually reversed. In this case, we actually see less of the 7-hydroxy, but more of the 8-hydroxy compound in this case. And the relative distribution of the S isomer, the two compound in S isomer, is also become fairly equivalent as compared to the case without induction. This is a 4-to-1 ratio. And you can see how the metabolic induction can impact the metabolism profile. Of course, since we have an impact on metabolic profile, of course, that will have an impact on toxicity. And this is not difficult to understand. Depend on different role of particular metabolite in the toxicity of compound, the enzyme induction may increase or decrease or cause no change in the toxicity of particular chemical. If a metabolite, for example, if a metabolite is responsible for the toxic effect of a compound, the induction of the enzyme can increase the toxicity. If there's only one route of metabolism elimination, depend on this, there's only one rate of metabolism on single metabolite that will increase rather than the total amount. And on the other hand, if the resulting metabolite is not involved in toxicity, there could be no effect. And also, if the resulting metabolite, even though formed at a higher proportion, if the rate of elimination can also be increased simultaneously, that can also lead to overall relatively small impact on the overall toxicity profile. If the foreign compounds is metabolized by several routes, then the induction may alter the balance of these routes, as we can imagine as well. Mechanistically, the induction of enzyme can happen at various levels, either at the mRNA level or at the protein level, or even at the protein degradation level. The most well-studied effect or mechanism of induction, this is lead to overall more synthesis of mRNA molecule of the encoded protein. And this is best documented in the case of the binding to aryl hydrocarbon receptor and the subsequent expression of the induced gene, such as cytochrome P450 enzyme. After binding to aryl hydrocarbon receptor after exposure to PAH series compounds, the induction of cytochrome P450, the expression of cytochrome P450 enzymes can be elevated under control of so-called xenobiotic response elements. So we'll see some examples here in a minute. Many P450s can be induced after exposure to polyaromatic hydrocarbons, and these are considered the receptor, the recognition factor of those polycyclic aromatic hydrocarbons are known as aromatic aryl hydrocarbon receptors. These are considered xenobiotic sensors to adapt the living organism for their metabolic and detoxification actions. We'll cover this particular receptor-mediated toxicity, and we'll go into more detail next lecture on AHR. But just to give you a flavor of it, these are basically ligand-activated transcription factors that will lead to series response in the nuclease and the expression of subsequent genes. And in this case, we're talking about key metabolic genes such as P450. So aryl hydrocarbon receptor, these are a series of proteins in the cytosolic fraction. And we'll have a diagram on the next slide. But their well-known substrate are polycyclic aromatic hydrocarbons or other aromatic carbon structures, such as TCDD, or also known as dioxin. AHR, these are proteins that are pretty widely expressed in most tissues, with high expression found in placenta, lung, heart, pancreas, and liver. In the absence of ligand, these aryl hydrocarbon receptors exist in the inactive complex, and upon sensing or binding of the available substrate, in this case TCDD, these protein factors are actually released from those inactive complex and transported to the nucleus and activate the expression of certain genes. In this case, cytochrome P450 genes. Of course, this is just one known pathways for the exposure of chemical causing subsequent enzyme induction by changing the gene level, the expression of certain genes. There are also other transcription factors other than AHR to induce cytochrome P450s, such as glucocorticoid factor or NRF2 factor that's also been shown to be able to activate the P450 gene transcription. Mechanistically, there are also other ways to regulate P450 in addition to the expression of mRNA level. So, for example, P452E1 protein, the main mechanism of regulation of that protein is actually through the stabilization of xenobiotics, such as ethanol binding to 2E1, acetone binding, pyrrole or isoniazid binding of the protein will cause a stabilization effect of that protein so that you will essentially increase the half-life of that protein and hence the bioavailability of that protein. Next slide, we'll have a diagram showing you how exactly the binding of dioxin, in this case, induce the inducer. This is not exactly dioxin without those chlorines, but this is just as a model to show one of the substrate for AHR. And you can see AHR initially in the cytosol exists in the inactive complex, the complex with several other proteins such as aryl hydrocarbon receptor interacting proteins and other chaperone proteins such as heat shock protein 90. And upon binding to its native substrate, it will be released from this inactive complex and transported to the nucleus. Upon entering the nucleus, aryl hydrocarbon receptor will bind to other interacting proteins such as AHR nuclear translocator, and the formation of that complex is believed to induce the gene expression by binding to the promoter regions of the cytochrome P450 genes. We'll go in more detail when we talk about the receptor-mediated toxicity. But this general theme of induction, this has been found to be true for several P450 enzymes, including cytochrome P450 1A1, 2B6, 2C9, 3A4, and others 4A families. They all seem to follow this general pattern after exposure to these polyaromatic hydrocarbons. The activation of those genes have been observed. Of course, if enzyme can be induced, they can be inhibited as well. In this case, we want to touch on briefly also about enzyme inhibition. And this simply means their activity can be suppressed in the presence of these inhibitors. In contrast to enzyme induction, inhibition usually only requires a single dose or exposure. Although the environmentally it may be of less of consequence than induction, it is probably of greater importance because a single dose will reduce the activity of the enzyme. Many of the different enzymes involved in the metabolism of fluorine compounds may be inhibited with consequences for biological activity of those compounds. As with enzyme induction, the consequence of inhibition will depend on the role of metabolism in the mechanism of toxicity or other biological activity. In the table below here, we can see these are well-known inhibitors for some of the common P450 enzymes, as well as other metabolic enzymes like esterase, aldehyde dehydrogenase, and xanthine oxidase. One of the examples here is grapefruit juice, which is a well-known inhibitor of some of the ion transporters, as well as P453 and 4. We have a couple of slides explaining that, but oftentimes if you pay attention to the drug label, oftentimes you will see that do not take this drug together with grapefruit juice or within certain time window, right? Because these are well-known inhibitors of P450 enzymes. Now, when we talk about enzyme inhibition, so we will need to quantitatively describe the reduction of enzyme kinetics. So this is just a review, hopefully, to you in terms of the enzyme inhibition and different qualitative measures to define the inhibition. So in general, enzyme inhibition can include specific and non-specific inhibition. And within the specific inhibition, it can also be classified into two general categories, with one being reversible and the other one being irreversible. And we'll focus on mainly on the reversible inhibitions here. And this can be further classified into so-called uncompetitive, competitive, and non-competitive inhibitions based on the different modes of binding to different components of the enzyme. And quantitatively, oftentimes this goes back to enzyme kinetics in biochemistry. And oftentimes we have certain kinetic parameters to define the rate of the enzymatic reaction. In this case, on the left, we have this Michaelis-Menten kinetics, which most of the enzymatic reaction follow, although there are certain exceptions that enzymatic systems do not follow the Michaelis-Menten kinetics. But in general, many cases we do see most of the enzyme follow this Michaelis-Menten kinetics. In this case, we have the Vmax and Km as important measures for the enzyme kinetics. The Vmax and Km can also be extracted from a different form of functioning here. On the right, this is showing the Lineweaver-Burke plot. And historically, this is useful when you want to plot many of these kinetic data manually. And this linear regression tend to be easier in terms of manual plotting on a graphic paper. This has been popular, especially before the availability of those graphing softwares. So just to briefly touch on these different binding mode and what does competitive and non-competitive, uncompetitive means. So competitive inhibition, this means that the inhibitor binds to the enzyme only. So in this case, you can see it binds to the enzyme rather than binding to the enzyme substrate complex, which is believed to be a important intermediate in the enzymatic reaction. So this is the general description of enzymatic reaction in here. You can see once the enzyme substrate encounter each other, they will first form this ES complex. And this is oversimplified kinetic model. But after the chemistry conversion of the enzyme, this will be further, the product will be released, right? So converting the substrate to product and released from the enzyme. and the enzyme will be ready for another enzymatic turnover. So the so-called competitive inhibition simply means the inhibitor will only bind to the enzyme, but then does not bind to the ES complex, which is different from the subsequent cases. In this case, uncompetitive inhibition, this illustrates the case when inhibitor only binds to the ES complex, forming an ESI. So many cases, the binding of this particular inhibitor can occur at a different side of the native substrate, right? So because the enzyme adopts a 3D structure, and other than the active site, there are other portions of the enzyme potentially can interact with an inhibitor, depending on different chemistry and the different inhibitor design. And in particular, for larger protein complexes, the inhibitor can also bind to other portion of that ES complex. So this is what we call uncompetitive. And lastly, what is called non-competitive is that you can see that binds to both enzyme and ES complex. In this case, the binding of the inhibitor will be much less selective compared to the first case. In this case, we call it non-competitive inhibition. Specifically, when we measure the enzyme inhibition, quantitatively, these are the important parameters to measure the inhibition quantitatively. One of them we call it inhibitory constant, Ki. And if you plot this in a hyperbolic plot, the half the maximal inhibitory concentration is what we call the IC50. So this is Ki, and the corresponding concentration we call the IC50. Now, Ki is a measure of the binding affinity, whereas IC50 is more reflective of the functional strength of the inhibitor. So IC50 oftentimes is used, you will see used more or reported more in the literature in terms of comparing the inhibition strength or functional strength of an inhibitor. In terms of different mode of inhibition, we talk about those terms competitive, uncompetitive, and non-competitive. Their calculations are slightly different, which is documented here. If you need it in the future in your research, this should be a good place to further investigate those actions. In terms of P450 inhibition, this is actually quite well studied field, and we know P450 in terms of its active site. This has that tetrapro ring, porphyrin ring, with the iron heme in the center. And for heme proteins, many times we'll see in P450 proteins, we'll see cysteine as the distal ligand, and on top there's a histidine ligand, oftentimes chelating the iron. And the binding of different ligands, this again depend on the electronic state and the interaction with the iron heme center in there. And some of the reversible complex or reversible inhibitors, including cyanide, carbon monoxide, nitric oxide, and other heme coordinating inhibitors like pyridine, imidazole, triazole. These are all nitrogen-containing rings that's similar to the side chain of histidine that, in a way, that also can serve as the coordinating ligand for P450. So these ligands iron center binding can occur reversibly. Hence, these are all known inhibitors. And you can, if you recall, the CO binding, the reduced spectrum of the CO bound form of P450 shows a signature absorbance at 450 nanometers. This is also the how P450 got its name, by having the signature absorption compared to other heme-containing proteins. So the binding of those, as you can imagine, of those ligands can inhibit the binding of other native substrate, for example, also binding of the oxygen in some cases, and will inhibit the P450 reactions. A number of foreign compounds are also known to basically destroy the activity of P450 enzymes. For example, halogenated alkenes, hydrazines, and compounds containing carbon-carbon double bond and triple bond. They all known to react with P450 and then bound to P450 in an irreversible manner, in a sense to inactivate the enzyme. One of the most well-documented example is the grapefruit juice effect. If you look at the drug label many times carefully, you will see the warning of not taking the grapefruit juice within certain windows of drug where the drug is being administered. Grapefruit juice has active ingredients that can block certain ion transporters, such as organic anion transporters and MRDs, MDRs, multi-drug resistant protein 1. So the induction inhibition of those important ion transporters will either reduce the bioavailability of active pharmaceutical ingredient or increase the bioaccumulation of some of the toxic drugs, some of the toxic drug metabolites. Grapefruit juice is also a very well-known documented example of P450 enzymes, in particular 3A4 enzyme. And there are studies have shown that the some of the active ingredient in the grapefruit juice have result in toxic metabolites that can have covalent binding and irreversible inhibition of P450-3A4 enzyme. Shown here are two examples. One is called bergamottin, and the other is called 6,7-dihydroxybergamottin, with the latter being one of the product of bergamottin. But these are two known active ingredients of grapefruit juice. Their presence in the grapefruit juice can lead to the bioactivation of these compound in terms of forming these epoxide compound. So this is being one of the reactive metabolites, and the other is the ring open form of that. So both form, these contain highly reactive entities, one being the epoxide, one being that you have these microacceptors, or in this form, microacceptors in this compound. There has been studies showing that these reactive metabolites can form covalent adducts with P450 proteins. Once formed, so they are essentially bound to the active center of the P450 enzyme and cause inactivation or inhibition of that particular enzyme. So overall, the presence of these compounds and reactive metabolites can reduce the biological level of P450 enzyme, causing enzyme inhibition. Last few slides, I want to touch on the species difference in metabolism and distribution and toxicity. These are also well-documented examples in terms of species difference. And the implication of this many times is on the extrapolation of some of the animal data and the evaluation of use of the animal data in terms of predicting human toxicity. Although many cases, animal studies have shown a good prediction in terms of metabolism toxicity, there are well-documented cases that document the distinct species difference of certain compounds. For example, neurotoxin, MPTP. There's a good story behind the MTP, and I'm going to share with you a video later this class for you to watch, perhaps next week. And this is a well-known example that the compound does not cause toxicity in rat, but rather leading to toxicity in humans. There are many aspects of ADME, these in terms of species difference. For example, simply based on absorption, human skin is generally less permeable to chemicals than that of some of the experimental animals, such as rats, mice, and mice, and the rabbits. And you can see some of the data are shown here in terms of relative rate of the skin penetration. You can see we have relatively low rate ranges to relatively high rate in the taking compound, in the taken up or the penetration of organic phosphate compounds. Other examples include pH. Of course, we know pH is an important factor affecting the ionization of the compound, and therefore the absorption and bioavailability of the compound. And you can see in terms of the human, the difference between pH of different media. You can see pH of the saliva, the difference between the species from man to sheep, and in this case, pH of the gastric juice. In human, this is extremely acidic, and you can see some of the extreme example, more basic at the end of the spectrum. This is found in sheep. So clearly the species difference exists. And in terms of biodistribution, this is another good example showing many times this is due to the plasma protein concentration and variation among the different species. You can see that some of the extreme cases include 20 milligram of protein in the plasma compared to in certain fish, compared to fourfold of that in cattle. And you can see here, we're just using sulfonamide as an example to illustrate the difference. These are sulfur-related group of antibiotics, sulfonamides, which are used to treat bacterial infection and some fungal infections. And you can see here that in terms of distribution, we can also see a wide range of distribution among different species, right? So human, cat, human here, and chicken lowest, human here, chicken lowest again, and in this case, the last case, the mouse is the lowest. Species difference in biodistribution due to the varying plasma protein concentration level. Of course, there are cases in difference in excretion. Many times this is due to the pH, as well as the volume and rate of production of urines. as you can imagine, this will lead to clear difference in the availability of the compound or excretion of the compound. And this is renal excretion, kidney excretion, same thing for bioexcretion. And lastly, metabolism. There are well-recognized example of metabolic rate difference of human enzymes and animal enzyme. In this case, we're looking at these different heterocyclic amine compounds and its metabolism by P451A2 enzyme. And here below, 3 graphs, you can see the side-by-side comparison of the human enzyme versus the rat enzyme with respect to each compound, and the plotted, what we call the mechanism-Lenten kinetics, clearly illustrate in terms of the turnover rate, as well as the Km value, their dramatic difference among the species. As a summary, as we can see this class, we discussed several factors, starting from the chemical factor, and we talk about the size, hydrophobicity of the compound, as well as structure, stereochemistry, and how that impact the metabolism and toxicity. Biological factors include genetic factors, which we focus our discussion on this time. But in addition to that, also depend on hormone influences, disease, pathological conditions, age, sex, and stress condition level, and diet, dose, environmental exposure, of course, because we're oftentimes talking about exposed to millions of compounds throughout the lifetime for in each individual. The expression of some of the biotransformation enzyme may be induced. We talk about enzyme induction. In particular, we talk about a P450 induction mediated by aromatic arohydrocarbon receptors. And we also talk about the inhibition of P450 in certain cases. Lastly, we talk about the species difference. And clearly, we saw examples that different animal species that can vary dramatically in all aspects of ADME. All right. Thank you for your attention. And this is the end of our class. I'll see you next time.